Okay, so this is the sort of the last bit of the things that I wanted to say, and I wanted to sort of put some of the things that I discussed in the last 3 lectures together today, and I'll use slides, so therefore, it might go a little fast. Okay, so I introduced to you yesterday quantitative genetics, and we already learned about polygenic traits from Shweta's lectures and my lecture, and before that we did in a couple of classes, in a couple of lectures, we talked about population genetics. So I want to talk about polygenic adaptation and the dynamics of that in large populations. So I'll explain as I go. So this is some work that is been done over the years with Wolfgang Stephan, who is at LMU Munich, and Archana Devi, who is now at Arizona State. Okay, so this is a little bit of revision of what we discussed in the last couple of, in this week. So I want to understand the phenotypic variation in terms of the underlying genetics, okay? And what is this underlying stuff? How do we characterize it? So there is this term used, genetic architecture, and one of you was asking what is meant by genetic architecture. So it's a bunch of factors, and this is not an exhaustive list. So for example, you can ask how many genetic loci are controlling or influencing that trait? What is the size of that effect? What is the frequency of these genetic variants? How does it interact with the environment? That means if there's a covariance and so on. So there's a long list. So if that's something interests you, you can look up this review article. Okay, so we want to understand the phenotypic variation, phenotypic dynamics on the basis of this underlying genetic details. So we already discussed about monogenic traits. One of the classical examples of that is lactase persistence in humans. So depending on the geographic region, there is one locus to about 4 or 5 loci. So it's monogenic to oligogenic trait. And if there's a domestication of these cattle, then in times of food shortage, it's good if you can use this new source of food. So it's a beneficial allele which will allow you to digest milk, and that is supposed to have arisen in certain parts of the world and had swept. So this again you have already learned this week, which is that mutant frequency, if it does not get lost, the mutant does not get lost, it may sweep through the population. That is, there could be large, rapid changes in the allele frequency. And Shivansh has already nicely explained to you this very same figure. And because sweeps, why are we so obsessed with the sweeps? Because they're very useful. They're very good signatures. We can use that to find the position of in the genome where the selection has happened, and what's the amount of selection that has happened. And he has already explained, you can use Taji-Must-D, LD, variability. So variability reduces, LD increases, Taji-Must-D also decreases, right? So this is all the stuff that you have already learned in this week, okay? So this is for, however, traits that are controlled or influenced by a few loci. More generally, this is covered by the population genetics theory, in which we deal only with genetic changes at the genetic level. But there's a problem in the sense that we know that selection acts on phenotype, not on the genotype, right? So therefore, but the way we do population genetics is that we don't worry about how it's coming from phenotype. We just assign the fitness directly to the genotype. For example, this concept of fitness landscape. So that's only in the genotypic space. So why is it rugged? Why is it one peak and so on? How is it connected to the phenotype that we're studying? We don't really care about it. We just assign it directly in most cases, okay? And another thing is that in population genetics theory, the focus is on new mutations. Again, this is a typical case, but there are some theories which also have started to take care of standing genetic variation. And so, as I already said, I'm just repeating this point, that the picture in population genetics theory is that a new mutant arises, then it might sweep through the population, then another one comes, it may sweep through the population, and so on. So this is an adaptive walk. And correspondingly, if I know how to connect the genotype to phenotype, the phenotype also changes. Okay, so that's the realm of population genetics. Now let's go to the other side, which is the polygenic architecture. So here, something like this you saw in Shweta's lecture, but this is not exactly the same. I'm not showing you the Manhattan plot here. What I'm showing you here is the heritability as a function of this chromosome positions. So let's focus on schizophrenia. So you see there are teeny blips. These What it means is that each of these guys are required to explain the heritability of schizophrenia. There are lots of them. About 70% of this huge region is required to explain the heritability of this. So it's a highly polygenic trait, okay? And all of them are tiny. The effect sizes are very small. For other diseases like this one, there are some large effect loci, and then they contribute to heritability. But something like schizophrenia, lots of small loci are required. Okay, so that was the polygenic architecture example, and this is traditionally studied in using quantitative genetics, which deals only at the phenotypic level, but it does not tell us what's going underneath in the genetic level. And there, of course, because we're dealing with phenotype, we assign the phenotypic fitness, and typical focus is on the effect of standing genetic variation, the object that I discussed yesterday, that is VA, the additive variance. Okay, so the picture here is that we have lots of small effect loci, lots and lots of them, and each of them changes tiny bit, such that still phenotype changes. So that's the sort of picture. Okay, so slightly more detail I'll give you. So I was trying to describe to you yesterday the infinitesimal model of Fisher, which to just repeat this point, which says that allele frequency, let's say like this one, it changes by little of them. Each of them changes only by little, but the genetic variance is assumed to be constant in time. Okay, so that was the underlying genetic architecture in quantitative genetics. That's what it implicitly assumes. Okay, so just to summarize what you have already learned, there are these two modes of adaptation. One in which we have subtle changes in allele frequency and driven by the standing genetic variation, the variation which is already there in the population. The other limit, the extreme, is the one in which you have this large rapid changes in the allele frequency, that's the sweeps, and this is driven by new mutations which will come, arise, and sweep through the population. So these are these limiting cases, and of course, in general, you will have mixture of these things, right? Okay, so the question that I'm asking here is, are there other adaptive scenarios that are possible? So we spoke about only sweeps, like in popgen picture, or only subtle shifts, which is in the quantitative genetic picture. Can we have both? Can there be an interplay of these two? Can both of them together in some fraction drive the adaptation process? I said for infinitesimal model, it assumes that the genetic variance does not change with time. Can that also change? Okay, and if so, in what parameter regimes? What should I change in this more general sort of setup in where we will see more general dynamics? And we would like to think of putting population genetics and quantitative genetics into a larger framework, because both are about adaptation, right? So we would like to put this in a single framework and study that. So this is what I do. I like to study general models. So specifically for this case, I would like to investigate general enough models that allow us to explore various types of genetic architectures and see what other scenarios come out of this, okay? And one thing is that the models that I like to deal with, we of course do numerical simulations like Wright-Fisher process and so on, but we also like to get little formulas. So that's what is called analytically tractable, meaning you want to get little formulas, okay? So this is the work that I've been doing on and off over the last few years, and you can find some of that summarizing, or you can find details in these papers. Okay, so how can we build up this larger model? So we can start with the phenotypic fitness, because as I said, selection acts on the phenotype. Then we can have a genotype-phenotype map. We discussed one of such map, the additive map, yesterday. So we can have a map which will bring the phenotype to the genetic level. Now at the genetic level, I do my popgen. I put in drift, mutation, selection, and so on, work out everything about allele frequencies. From these genetic details, I go back to phenotype through this map, and therefore complete the cycle and make predictions about phenotype. Okay, so that's the sort of program. Okay, okay. So I'll now describe this specific model, and this is not some model that we cooked up. It's been, there's sort of in some ways, it's a very old model actually, due to Wright and various papers, including us, have worked on various aspects of it. So let's now implement this model, this little framework that I said we would like to put in this model. So I'll start with the phenotypic fitness. I specify that, and I'm thinking of height as an example trait. So as you know, as you expect, that there's probably an optimum height for which individuals have a high fitness. Very short and very tall perhaps have a low fitness. So that's called a stabilizing selection, which has this, which is modeled by a Gaussian or a normal function. Okay, so that's what I'm assuming here. So I've defined my phenotypic fitness for you. Okay, then I need to go from the phenotype to the genotype. And yesterday we learned about P equal to G plus E, how we connect phenotype to the genotype. That's this map. And yesterday we assumed an additive map, and that's what I'm going to assume because I'm thinking from the human popgen, human quantitative genetics, human population genetics, for which additive map is a good approximation. So just to remind you again, if I have a genome of an individual which has a T allele at the first locus, S, which is a short allele, a second locus, and so on, then additive map means that tall allele increases, has the effect to increase the height by an amount gamma, whereas short one decreases by an amount by the same amount, perhaps minus gamma. Now, remember what we said yesterday, P equals G plus E. G is the addition of these gammas, whereas E in this environment, which was the stochastic component, right? So these gammas in general are different at different loci, but these gammas are now random variables. So I'm adding the environmental effect by modeling these gammas, the effect sizes, as random variables. They're chosen from some distribution. But let's not worry about it. But this is something, again, to connect with what you learned yesterday. Okay, so we have gotten from our phenotype fitness I have defined for you. I know that if you give me any genetic sequence, I know what its fitness is. And then now I can put in mutation, drift, recombination, whatever is relevant for that problem. Specifically, I'm looking at today, I will model it using the Wright-Fisher process that we learned in the first lecture. So again, just to remind you, so I'm thinking of n individuals, and let's say for simplicity, there are just these 4 individuals, and as we said, because selection, maybe this has, because I said it's a stabilizing selection, so it's likely to produce less offspring compared to others. And then we do random sampling too, because of finite carrying capacity. And then we could also have small to S to T or T to S mutations, and that's how we will form the offspring generation. Okay, so this is the generalization of this little Wright-Fisher process that we learned in lecture 1. I'm demonstrating here, I'm writing this here, assuming this is asexual haploids, but actually what we do is to look at recombining populations, which have a lot of recombination, and these are diploids. Okay, so basically this kind of models one can write down. I did not emphasize it in the other lectures, but these are all the stochastic models which are of course relevant. But a good starting point for our intuition is to just neglect drift completely. So these are called deterministic models, and it assumes that the population size is infinite. So we can completely neglect drift and understand those models thoroughly, then turn on drift, and then see if the same intuition goes through or what new effects drift might bring. Okay? Okay, so just to show you what I do, if I'm thinking of deterministic ones, that means ignoring the drift, I need to write down certain bunch of equations, which we will not worry about. But I saw there's some people who are familiar with nonlinear equations. So we need to write, in this model, it turns out you have to write some coupled nonlinear equations. Such equations, if you have dealt with, you know they are almost impossible to solve exactly. Okay? So what we try to do is to therefore develop approximations, sensible approximations, so that it gives us some insight into what's going on. So that's one model that we studied some time ago. We also include drift, so therefore I now need to write down some bunch of equations for that. So as I mentioned, that is dealt using what is called a diffusion theory. Again, don't worry too much about what's all written here, but again, those who deal with these things, you have to write down certain partial differential equations, and then these are also impossible. I mean, very, very extremely hard to do anything exactly. But then you develop some intuition, some approximations to get some simple formulas for it. Okay, fine. So this I've defined for you the philosophy here. That is how we will connect these two phenotypic and genotypic level. I've taken a specific model and I told you the methods that I use. So now let me just tell you quickly what we find. So the specific situation I'm thinking of is that I have this phenotypic fitness. It's in the we have a population which is under stabilizing selection. So therefore, the phenotypic fitness is our favorite normal function. Okay, and there's an optimum here. There's an optimum height at which the fitness is maximum. And then let's say we assume that there's a sudden shift in the optimum. So that now the phenotypic fitness is this dark blue one. Okay, so the population initially is sitting over here, right? So the ones which have this height initially, the gray curve, they have a low fitness, because now the optimum has shifted, right? So now this population has to adapt, and we want to know what are the dynamics of this adaptation process. So this is the mean which increases quickly, okay? So before I say a bit more, I need to tell you one more thing about the genetic architecture of the polygenic trait I'm thinking about. So I told you that I'm going to think of both in large populations, very large populations, as well as large populations that have drift. Furthermore, you have heard about these small effects, large effects in yesterday's Shweta's lectures and my lecture. But I also told you, I emphasize this repeatedly, that when we say something is weak or strong, small or large, it's always relative to some scale. Okay? So don't forget that. So when we say small effect, large effect, what does that mean? How do we define that? It turns out that for large populations, whether you have drift or not a drift, we can define a critical effect size below which, if the effect is below it, we call it a small effect. If it's larger than that, then it's called a large effect. Okay? So it's not some arbitrary classification, rather the things are classified on the basis of an objective parameter which depends upon n, mu, s, and so on. Okay. So again, I will not give you any formulas, but what I do want you to remember is that when I say small effect loci, it's synonymous to being polymorphic. Large effect are the ones which are monomorphic or close to fixation. So just remember this because I'll be using small and large in that sense. Okay, so let me just quickly give you all the answers. So let's first start with a simple model in which there's no drift and most effects are small. That means there's a lot of genetic variation because I said these are small effects means polymorphism. So there's already a large initial genetic variance, lots of things to choose from, and therefore when adaptation occurs, it can use that initial genetic variance to adapt the population. So it turns out that there is no change in the genetic variance in this model, and at the allele frequencies, there's only subtle shifts in the small effect locus. So these small effect ones are the ones which are polymorphic, and there's only small change in that. Genetic variance does not change. Both the things that we saw in the context of quantitative genetics. I said most effects are small. That means some effects are large, like this figure that I showed you earlier. Let me just go back. Yeah. So there could also be some large effects, lots of small effects. Some large effects are also possible, right? So let's think of that. So what happens? So the question there is, if you have a large effect locus, what happens there? It's a monomorphic one. It's near fixation. Can there be sweeps at these large effect loci? So that's the question, and the answer is not obvious, okay? But using a simple model and a more detailed model, what we find is that sweeps do not occur. Unless you take some extreme parameters like you shift the optimum a lot and so on, we don't see any sweeps in this model. So the point here of this little slide is that if I have a very large population and most effects are small, the behavior is essentially the same as the infinitesimal model or the quantitative genetics. Okay, so this is the old scenario. So one good thing is, one thing that whenever you do modeling, you should be able to reproduce what's known. Then only you have a complete model, right? That should come out and something more. So this is the, what we know already is there already in the model, but let's see if there's something more. Yeah, please.
So ma'am, is it also associated with the genetic structure because the genes are large, so they are not linked? Means during recombination, they are just getting away. That's why they are causing no sweeps.
So I'm assuming a lot of recombination. in fact, I'm assuming complete linkage equilibrium in this model.
Okay.
Including linkage equilibrium is hard in these models. Okay, so what the story I'm telling you is completely linkage equilibrium. And the reason we don't see sweeps is because there's so much other stuff. You can make it with this teeny things, just like the quantitative genetics picture. So that's why we don't see sweeps. But when we actually work out in this, in these models, there's two competing factors, and it's not obvious, therefore, whether sweep will occur, whether it matters. It does occur, but only if you make extreme changes. Okay. So it's nothing to do with recombination, because recombination, I'm assuming, is infinite.
Thank you.
Any other questions so far? Okay, fine. Something new. So no drift, let's say, and now most effects are large. That means we have these alleles, these loci, where frequencies are either 0 or close to 0 or close to 1. Okay, so now because they're close to 0 or 1, there's hardly any variation in the population to start with. So now this population has to adapt. The variance has to increase, right? Which is unlike the infinitesimal model. So in fact, that's what we see. There's a dramatic increase in the variance. Okay, so this is unlike what we know from infinitesimal model or quantitative genetics. Besides, because variance is increasing, so initially things are sort of monomorphic, low frequencies, right? Variance is increasing means what? There must be some polymorphism. Things are going from frequency 0 to frequency 1. That means at many of these large effect loci, there are lots of sweeps happening. So this regime is characterized by increase in genetic variance and lots of sweeps. Okay? Okay, so that was a very quick summary of very large populations. Let me now let in drift. Okay, so this is a complicated stuff, don't worry too much. But what I want to say is that if I have finite population, we spoke about mutation and drift balance in one of the tutorials, but now I have mutation, selection, and drift balance. So all three I have to consider. So let me first start with asking what happens at that balance, and then we will talk about adaptation dynamics. And I particularly wanted to show you this slide because you guys can understand this because you have done the tutorial. Okay, so this is what we find. So so we analyzed this, Archana and I, and then we looked at distribution at a locus, which has this qualitative feature. Weak mutation, remember, weak means relative to something. So when I'm saying weak mutation, it means relative to drift, it's weak. Everybody recognizes this now. When the mutations are weak, you have near fixations. Remember, I told you about this beta distribution. U-shaped distribution is what you were supposed to get, right? So that's what happens in this more complex model also. So when the mutations are weak, then you have this U-shaped distribution. But in this parameter regime, when we are thinking of adaptation, this is the work of another group, they find that initial genetic variance is small, okay, but it increases, not maybe too much, but it does increase. And however, there's no sweeps in this case either. But if you have strong mutations, which is what I'm interested in, and I also have strong mutations, and we can have strong selection and weak selection. So now, in one case, when we have strong mutations but weak selection, then we have a unimodal distribution, again the kind of thing that you did, where you neglected selection completely, right? So remember we said for n mu larger than 1, you will have a Gaussian sort of thing and a U-shape. That's exactly what I'm showing you here, okay? Well, not exactly, but something like that, qualitatively. Okay, so when we have strong mutation but weak selection, we have this unimodal distribution. But if I have strong mutation and strong selection, then this distribution becomes bimodal. Strong selection, strong mutation means then I have to ask which one is stronger, relatively, which one is stronger. So this bimodal distribution comes when both are strong, but selection is stronger than mutation. Okay? So in that case, this unimodal distribution becomes a bimodal. So since you have thought about the unimodal case and the U-shaped case, I think you are in the position to think about how this bimodal distribution might be coming. And I strongly encourage you to think about it when you go back home. Okay, so now we are in the regime when we have a finite population, and we see that for small effect loci, we have a unimodal distribution, but for large effect loci, we have a bimodal distribution. Okay, that was in the stationary state, which is at the balance, but now let's look at the adaptation dynamics again. So here we find that the genetic variance does not change. It remains roughly constant. What about allele frequencies? Small effect loci, which are like bimodal, which are these lots of them shown here, they go to teeny changes as they should. They're polymorphic and they go little changes. But for the large effect locus, I have a bimodality. That means some allele frequencies will be of low, at low value, some will be high. So we could have something going like this, right? Or in which case, there's a subtle change in the allele frequency for small effect as well as the large effect locus. This is also possible, that small effect ones do small changes, but the large effect one, it started over here, and as time went on, it moved here. Okay, so that's this blue one. That is a sweep. It's not the hard sweep, it's a soft sweep, but there's a large, rapid change in the allele frequency. Okay, so therefore we find in this case, in this parameter regime, which I have not completely clearly specified for you, but we find that we can have, we may not have sweep at large effect locus, but we can also have sweep at large effect locus. So when does sweep happen? That means what's the probability of the sweep to happen? So we can calculate that, and we find that while in the infinitely large populations, that means there was no drift, there was no sweeps, but now in the finite populations, the sweeps that were not happening for some reasonable parameters, now for finite population, they can occur. Sweeps prevented in the infinite population can occur in finite population, and this uses some ideas that Archana had developed, and this is from simulations, and this is her formula. Okay, so just to summarize this little part, selective sweeps, there is, people have thought about this question because, there's a long history. Quantitative genetics in the 1920s and so on, where you're thinking of subtle changes only in the frequency. Then 1970s, Maynard Smith and Haig, Levantine, you have all the sweep business. And now we're thinking that, okay, we know this part, we know that part. Let's see, can we have both, right? So in this full model, I mean, it's a sort of general model. There are various aspects of it, we will study by various people. The summary is that in infinitely large populations, selective sweeps are not seen. In very small populations, again, selective sweeps are not seen. But moderately sized population, which is given by this criteria, sweeps that were not happening, we find they can occur with some reasonable probability. Okay? And more broadly, I started with saying what other things can happen, what are the other possible adaptive scenarios. So we find that there is some parameter regimes where, just like in infinite model, we have subtle changes in allele frequencies and no change in genetic variance. That's the two key points in quantitative genetics which are assumed. But by studying this general model, we can now pinpoint that in what parameter regime, what new things you will see which goes beyond the standard paradigm. We can see sweeps and changing genetic variance, and when we have these parameters, we can see subtle changes and changing genetic variance, sweeps and constant genetic variance, when we have these other ones. Okay, so that's sort of the broad thing that I wanted to say. And can we see these sweeps? Can we catch these sweeps? The sweeps that we are talking about are soft sweeps, and they're notoriously hard to catch. Okay, so I don't know. What theory can tell you is that where perhaps to look. Whether you can see it or not, I don't know. So that still is a big question. Okay, so that's all I wanted to say. Thank you.